Now we will discuss homology modeling. Protein are biomolecule and macromolecule that comprise one or more long chain of amino acid residue. Protein perform a vast array of functions within organism, including catalyzing metabolic reaction, DNA replication, responding to stimuli, providing structure to cell and organism, and transporting molecule from one location to another. Macromolecule composed of one or more chains of amino acid. Each long chain of amino acid is called polypeptide. Each amino acid linked by a peptide bond. Protein play important structural and metabolic role in the human body. Let's see this figure. There are 22 types of amino acid. See this one. Protein structure consists of a primary. This is a sequence of a chain of amino acid. A secondary. This is because of hydrogen bonding of the peptide backbone of the amino acid to fall into repeating a system. Then tertiary, this one is three-dimensional folding pattern to side chain interaction, and the last is quaternary. This is related to interaction between amino acid and related to amino acid chain. Now let's see this figure. This is the structure and this is the process. Let's see this one. Primary, the process to assemble. A secondary, folding. Tertiary, packing, and the last quaternary interaction. There are two ways to determine protein structure. The first one is experimental. This experimental consists of X-ray crystallization. This method provides the maximum data regarding the tertiary structure of protein. X-ray are passed through the rotating crystal and diffract rays are collected on the target and analyzed by computerized system. Crystal quality is crucial in this case. Thus, it results in very detailed data on automatic arrangement within a rigid protein with other neighboring ion, ligand, and molecule, but may not work so well with a protein, which have lack flexible domain and therefore do not form precisely ordered crystal structure. They require relatively large amount of the protein in case of carry out this X-ray crystallization. Then the second one is nuclear magnetic resonance. We call that one as NMR. This method depends on the effect of varying radio frequency wave on molecular resonance of various atoms within the protein. It needs larger quantities of protein in a stable soluble form at room temperature and to remain stable for the long duration of data acquisition. And the last is cryogenic electron microscope. The method are a suite to study of macromolecule complex or even a cellular organelle, which are relatively bigger on the molecular scale and can help to reconstruct the tertiary structure of a single particle. This has the great advantage of being the challenging prerequisite of protein crystallization. Cryo-electron microscope is a variant at temperature at or below the of a liquid nitrogen and can visualize protein structure at a very high resolution. 2 is less than that of method like NMR spectroscopy and crystallography. Then computational. Homology or comparative modeling. The homology modeling is one of the computational structure prediction methods. They are used to determine protein three-dimensional structure from its amino acid sequence. Homology modeling is a procedure that generates the previously unknown protein structure by fitting its sequence or a target into a known structure. We call that one as a template. Given a certain level of sequence homology, at least 30% between target and template. Then fault recognition or threading. This is a method of protein modeling which is used to model those proteins which have the same fold as protein of unknown structure, but do not have homologous protein with known structure. The prediction is made by threading, for example, aligning amino acid in target sequence to a position in the template structure and evaluating how well the target fit the template. And the last of this computational method is ab initio prediction. Ab initio or de novo protein modeling method seeks to build a three-dimensional protein model from a scratch. For example, based on physical principle rather than on previously solved structure. The ab initio method is often prepared for a structure prediction when there is no or very low amount of similarity the very low amount of similarity for the protein. The protein are deposited in database. One of the larger protein structure repositories is a protein data bank, PDB, or call that one as a PDB, protein data bank. From the computational protein structure prediction, we obtain several methods such as knowing of the three-dimensional structure. In this case, will help in the rotational design of site-directed method, can be of great importance for design of duct, and greatly enhance our understanding of how protein function and how they interact with each other. Then we move to structural information from experimental method. In this case, will give or obtain much more slowly. Technique involve elaborate technical procedure, and many proteins fail to crystallize at all or cannot be obtained or dissolved in enough of quantity for animal measurement. And the last of this structural information from experimental method is the size of the protein is also a limiting factor for animal. Now let's see homology modeling. Homology modeling is a modeling a protein three-dimensional structure using a non-experimental structure, a homologous protein. In this case, protein as sharing a significant similarity of sequence can be expected to also share a significant similarity of a structure. The protein tertiary structure is better concept than amino acid sequence. Generally, the process of homology modeling involves four main types. The first one is a target identification. In a target identification that consists of database mining and a template recognition, then a sequence alignment. The third one is a model building. This consists of backbone generation and loop modeling. And the third one of this one is sidechain modeling. Then the last is model refinement. This model refinement consists of model optimization and model validation. Now let's see the step-by-step modeling, step-by-step homology modeling part. We'll start from this point, identify template structure, align a sequence to template structure, three sequence into the template structure, loop building and model building, evaluate the model, then model okay. If it is yes, then the process will in. But if the model is not okay, then we'll back again to this part. So let's see this one. We have the target sequence and we have a template. Then move to this sequence alignment. Then there's a homology model. The protein database is a collection of sequence for several sources, including translation from annotated coding. There are three main sequence database. There are three main sequence database. The DNA database of Japan, we call this one as DDBG, Embel nucleotide sequence database, this one, and National Center for Biotechnology Information, Gene Bank, this one. Now let's move to template recognition. Template recognition is a technique to identifying the proper template for the protein. This step aims to searching the related protein sequence as a template to the target sequence in any structural database of a protein. Also, determining the occurrence of model depend on the selection of proper template. For this step, pasta and blush can be used in this case. After the initial alignment and finding a structural concept region among templates, we choose the final template. This give a probable set of templates, but the final one is not yet decided. Sequence alignment is an arrangement or two or more sequence, highlighting their similarity or a way of arranging a protein or a DNA sequence to identify a region of similarity that may be a consequence of evolutionary relationship between the sequence. The sequences are paired with a gap or dash so that wherever possible, column contain identical character from the sequence info. Blast and crystal way are the tool for align the protein or nucleotide sequence. Now let's move to determining a structural concept region. In evolutionary biology and genetic, concept sequence refer to identical or a similar sequence of DNA or RNA or amino acid, in this case a protein. They occur in different or same species over a generation. This sequence show very minimal change in their composition or sometimes no change at all over a generation. Target protein is supposed to assume the same conformation in concept region. Let's see this one. In this case, we can see that this one, first step is building a model protein by homology, is determining what region are structurally a concept or constant among all these reference protein. Then the second, target protein is supposed to assume the same conformation in concept region. The structural building involves several steps such as backbone generation. Creating the backbone is trivial for most of the model. One simple transfer the coordinate of those template residue that show up in the alignment with the model. This step aims to assign the coordinate within the concept region. Then building a loop. For most homology model, a building case, the alignment between model and template sequence contain a gap. Gap in the model sequence are addressed by optimizing residue from the template. Gap in the template sequence are treated by inserting missing residue. The missing residue that into the continuous backbone. And next, conformational set for side chain. In this case, a library of common rotameter, the library of common rotamer extract from high resolution X-ray structure are often used to position a side chain. The various rotamer are subsequently explored and score with a variety of energy function. Then the last is a structure refinement. In this case, using algorithm to predict the correct rotamer and conformation. To predict the sidechain rotamer with high accuracy, we need to correct backbone, which in turn depend on the rotamer and their packing. First, we see this one. The common approach to address this problem is to iteratively model the rotamer and backbone structure. First, we predict the rotamer, then remodel the backbone to accommodate rotamer, followed by a round of repeating the rotamer to the new backbone. This process is repeated until the solution converge. Then now let's see several homology modeling tool. We have this one, a Swiss model, Raptor X, Spread 3D, Info, the CPF model server, and SDSCE. Every protein structure contains error, and homology model are no exception. The number of error in this case for a given method mainly depend on two values. The first one is the percentage of sequence identity between template and model sequence. The second, the number of error in the template. There are two principal different ways to estimate error in a structure. The first one is calculating the model energy based on force field, and the second one determining normality in these that describe how well a given character of the model resemble the same character in the real structure. The Ramachandran plot is a plot of the torsional angle P and C of the residue, in this case amino acid, contained in a protein. So I think that's all for this time. Thank you so much.